For centuries, human beings have basically been engaged in this all-out brute force war against our tiniest neighbor.
Absolutely.
Like, if you picture how we usually deal with mosquitoes or, agricultural pests, it looks a lot like blunt trauma. We use rolled up newspapers or we hang those electrified zappers on our porches.
Right. Or we deploy massive crop dusters to just blanket entire fields and clouds of chemical sprays.
Exactly. It's messy, it's indiscriminate, and honestly, it's a very primitive way to fight a war.
It really is. It's basically the equivalent of using a sledgehammer to swat a fly.
Yeah.
We've historically relied on that kind of overwhelming chemical and physical force just because, well, that was the only tool we had available to protect ourselves, from the diseases they carry and the crops they destroy.
But today, we are throwing away the sledgehammer. Welcome to the deep dive. We are looking at some really fascinating source materials today to understand a highly complex new frontier, which is basically weaponizing insects to eradicate their own species.
It's a massive shift in how we approach this.
It really is. So, just so you know, our mission today is to synthesize a stack of sources for you. We've got a report from the Harvard Chan School of Public Health on alphabets verily. We've got an ecological risk assessment from Oxford University.
We also have some regulatory documents from the US EPA.
Right, and real-time data out of Singapore, plus some scientific literature from Taylor and Francis. Okay, let's unpack this. We are moving away from traditional pest control and entering the frontier of biological engineering. So, to start us off, what is the foundational science making all this possible?
Well, the foundation of this first approach actually relies on something entirely natural. It's a bacterium called Wolbachia.
Okay, Wolbachia.
Right, and Wolbachia is incredibly common in the insect world. It naturally infects about 60% of all insect species globally.
Wow, 60%?
Yeah, everywhere. But here's the critical detail. It does not naturally occur in the Aedes aegypti mosquito.
And that specific mosquito, just to remind you, is the primary vector, like the main carrier for human diseases like dengue, Zika, and yellow fever.
Exactly. So we have this major disease-carrying mosquito that is naturally free of Wolbachia.
So how does introducing this, common bacterium suddenly turn the mosquito into a weapon against its own species?
Well, it comes down to a biological mechanism called the incompatible insect technique, or IIT for short. The Wolbachia bacterium essentially acts as a reproductive saboteur.
A saboteur. I like that.
Yeah. So scientists at Verily, which is a subsidiary of Alphabet, they have this project called Debug. They actually take Aedes aegypti mosquitoes and infect them with Wolbachia in a lab.
Okay.
Then they rear millions of these mosquitoes, sort them by sex, filter out the females, and release only the infected males into the wild.
And I should clarify for you listening, this release is safe for humans because male mosquitoes do not bite.
Right.
They don't feed on blood at all.
No, they only feed on flower nectar. Only the females bite us because they need that protein in our blood to develop their eggs.
Right, so the released males are completely harmless to you and me.
Exactly. But when those Wolbachia-infected males fly out and mate with the wild, uninfected females, a biological firewall kicks in. It's called cytoplasmic incompatibility.
Cytoplasmic incompatibility. Okay, how exactly does that firewall work? Like, what is the bacteria actually doing inside the mosquito?
Think of it as a lock and key mechanism. The Wolbachia bacteria actually alters the sperm of the male mosquito.
Oh, wow.
Yeah, it essentially scrambles the genetic password required for fertilization. So when that male mates with a wild female, her egg tries to read the sperm's genetic code.
But she doesn't have the bacteria.
Right. Because she doesn't have the exact same strain of Wolbachia to unscramble that password, the egg's code completely rejects the sperm. The wild female goes on to lay her eggs as usual. But because of that cellular incompatibility, those eggs will never, ever hatch.
That is incredible. It's like we are sending in saboteurs to distribute poison blueprints to an enemy factory.
That's a great analogy.
Like, the factory spends all its resources and time building the product, but as soon as it comes off the assembly line, the whole thing just crumbles.
That is a very apt way to look at it. You are hijacking their own biological drive to reproduce and turning it into a total dead end. And the data from our sources shows how effective this can be.
Right, the real-world trials.
Yeah, in isolated trials, it's huge. Like in a field trial in Fresno, California, researchers recorded a 95% suppression of the local mosquito population.
95%? That's almost total eradication in that area.
It is. And in Innisfail, Australia, they achieved an 80% suppression.
And we also have real-time data out of Singapore, right? The National Environment Agency there has been running Project Wolbachia.
Yes, and this is where it gets really impactful. Singapore reported a 66% decrease in actual dengue cases in early 2026.
Compared to the previous year.
Right, compared to the previous year in the areas where they deployed these modified mosquitoes. It's a massive demonstration of population suppression, leading directly to a reduction in human disease.
But, I mean, the logistics behind this are just mind-boggling to me. You mentioned Verily is rearing and sorting millions of these mosquitoes.
Millions, yes.
How can they possibly sort millions of tiny insects accurately enough to ensure absolutely no biting females slip through the cracks? Like, if the whole point is to release harmless males, what happens if the sorting machine makes a mistake?
What's fascinating here is the sheer level of engineering precision that this demands. You cannot just have a team of grad students with magnifying glasses separating them.
Right, obviously not.
Verily uses advanced computer vision, artificial intelligence, and automated mechanical sorting. The AI is actually trained to separate the sexes based on microscopic physical differences,
Like what kind of differences?
like the size of the pupa before they even hatch into flying adults. The males and females are slightly different sizes.
But still, what if a batch of females does get through? I mean, nothing is 100% perfect.
Well, if you accidentally release a significant number of Wolbachia-infected females along with the males, you change the entire outcome.
Okay, how?
Because those infected females can successfully reproduce with both infected males and uninfected wild males. Since they both have the same Wolbachia strain, the genetic password matches.
Oh, I see. So instead of crashing the population through incompatibility, you just end up replacing the wild population with a brand new lab-reared Wolbachia population.
Precisely. It is the fundamental difference between population suppression and population replacement.
Got it.
Both strategies do exist in biological control, but if your specific goal is to eradicate the biting pests entirely from a neighborhood, which is suppression, then releasing females completely undermines the technique.
That is a razor-thin margin for error.
It really is.
But our sources show this is no longer just some highly monitored science experiment, right? We are moving into nationwide strategy.
Very quickly, yes.
Yeah, in December 2023, the U.S. Environmental Protection Agency made this landmark decision. They approved a product called ZAP males.
Created by a company called Mosquito Mate.
Right, and these are Wolbachia-infected Aedes albopictus. That's the Asian tiger mosquito, right?
That's correct.
And the EPA approved their use across all U.S. states and territories. I mean, a business in Maryland has already released 600,000 of them. Why is there such an urgent push to scale this up right now?
Well, the urgency is largely being driven by climate change. As global temperatures rise and weather patterns shift, the livable habitat for these disease-carrying mosquitoes is rapidly expanding.
They're moving further and further into the U.S.?
Exactly. And because the Asian tiger mosquito thrives in urban environments, densely populated cities are disproportionately affected. This places a massive burden on public health infrastructure.
So we need a new solution, fast.
Right. The argument for this biological method is that without it, cities will be forced to dramatically increase their use of traditional chemical pesticides just to manage the growing populations.
Hold on, though. The EPA is the agency that regulates chemicals and pollutants. Are you telling me they're legally treating a swarm of breeding, flying insects exactly the same as a can of chemical bug spray under my sink?
If we connect this to the bigger picture, you can see how regulatory bodies are being forced to adapt old laws to fit entirely new scientific realities.
Okay.
The EPA regulates these mosquitoes under FIFRA. That's the Federal Insecticide, Fungicide, and Rodenticide Act. Under this framework, they legally classify the Wolbachia bacterium as a biopesticide.
A biopesticide, a living pesticide.
Exactly. Because the end goal of the product is pest control, the EPA applies the existing pesticide regulatory framework, but with safeguards tailored to a living organism.
What kind of safeguards?
For example, the ZAP males approval is strictly term limited. It automatically expires in December 2028.
Oh, so they have to review it.
Right. It forces a mandatory reevaluation of the data before they can continue selling it. Furthermore, the EPA mandates a strict quality control threshold. If the manufacturing process accidentally produces more than one female per 250,000 males, all distribution must halt immediately.
Wait, one female in 250,000 males. Who is actually counting 250,000 mosquitoes to ensure that ratio? Is the EPA standing there with the clipboard? Or are they just trusting the company's own math?
That is a very valid critique.
Because if it's the latter, that feels like a massive regulatory loophole.
It is a known tension in environmental regulation. The EPA relies heavily on the manufacturer's own quality assurance data and self-reporting, subject to periodic audits. It's definitely not an EPA agent inspecting every single batch.
Right.
Regulators are trying to balance the negligible risk of human exposure to a few extra mosquito bites against the really urgent public health need to stop disease vectors. But you are absolutely right to point out that scaling up biology is inherently messier than scaling up chemical production.
Yeah, and as fascinating as Wolbachia is, I mean, it is ultimately a natural bacterium that we are just exploiting. Our sources from Oxford University indicate that the science is already escalating to a whole new frontier, which is synthetic gene drives.
Yes. This is where we move from utilizing a naturally occurring bacteria to actively rewriting the fundamental code of life itself.
Using tools like CRISPR.
Right. Exactly, using CRISPR-Cas9.
I know this gets incredibly technical, but how exactly does a gene drive differ from normal genetics? Because in standard biology, traits don't just automatically take over an entire population.
Right, they don't.
Yeah.
Let's look at normal genetics, Mendelian inheritance.
Mhm.
Normally, if a parent has a specific mutation, there is only a 50-50 chance they pass it on to their offspring.
It's a coin flip.
It's a coin flip. A synthetic gene drive is designed to rig that coin.
How so?
Think of the insect's reproductive capability as a car. In a normal scenario, an insect might inherit a mutation that breaks one of its brakes. We call this a heterozygote. One working copy of a gene, one broken copy.
Okay, so the car has one working brake and one broken brake.
Exactly. It can still stop. The insect can still reproduce. But a gene drive doesn't just sit there. Once inherited, it actively seeks out the working copy of the gene from the unedited parent and smashes it.
Wait, it copies itself over.
Yes, it actively copies the mutation over, converting a heterozygote into a homozygote.
Yeah.
Now the car has two broken brakes.
Wow.
The insect's reproductive system is guaranteed to crash. The gene drive forces the genetic edit to spread to nearly all offspring, generation after generation, completely bypassing the normal rules of evolution.
Here's where it gets really interesting. It's essentially a biological computer virus written specifically to seek out and crash a pest's reproductive code.
That is an excellent way to synthesize it.
Yeah.
It actively copies and pastes itself across the genome of the target population. And scientists are exploring this not just for mosquitoes, but for massive agricultural pests, too.
Right. The Oxford paper highlights specific targets like the fall armyworm, the diamondback moth, and Drosophila suzuki,
Yes.
which is the spotted-winged Drosophila.
Right. And it is a devastating agricultural pest. Unlike other fruit flies that just go after rotting fruit on the ground, the spotted-winged Drosophila has a specialized ovipositor. It's a sort of saw-like appendage. Yeah, it allows the fly to literally slice into perfectly healthy, ripening berries and vine fruits to lay its eggs.
Oh, that's awful.
When the larvae hatch, they eat the fruit from the inside out, rendering entire harvests completely unmarketable. We're talking about billions of dollars in global crop damage.
So a synthetic gene drive could stop that.
Theoretically, a synthetic suppression drive could spread a lethal genetic trait through the entire Drosophila suzuki population, crashing their numbers without a single drop of chemical pesticide.
But the idea of a self-replicating lethal genetic code spreading through a wild population, I mean, that sounds like it could easily get out of hand. What stops this biological computer virus from just wiping out everything everywhere?
That is the crucial nuance in the Oxford paper. The highly contagious scenario you are imagining is what we call a low-threshold drive.
Okay.
It only requires a very small number of modified insects to be released, and it is designed to spread easily and widely, potentially across a whole continent.
That sounds risky.
The scientific community is highly aware of those risks, which is why they are also heavily investing in high-threshold or contained drives.
But how do you geographically or temporally contain a genetic edit that is inherently designed to spread?
One of the leading methods is called a daisy chain drive.
Daisy chain.
Yeah. Instead of putting all the genetic components needed to copy and paste the lethal trait into one neat, infinitely replicating package, scientists split them up into a chain.
Okay, I'm following.
So component A drives component B. Component B drives component C. And component C actually carries the lethal suppression trait.
Okay, so it requires all the pieces working together at the exact same time.
Exactly. And because of the way genetics naturally reshuffle over successive generations in the wild, those components eventually separate from one another.
The chain breaks.
The chain breaks. Once the chain breaks, the drive can no longer copy itself, and it naturally burns itself out after a few generations.
Oh, that's brilliant.
It allows for highly localized pest control, say, protecting a specific valley of berry farms, without the risk of a global genetic wildfire.
Okay, so we have these contained biological weapons. But what happens to the ecosystem around them? I mean, even if the drive burns out exactly as planned, we are still talking about fundamentally altering or even eradicating entire species in a given area.
Yes.
And the Oxford paper outlines what they call pathways to harm.
Right. And these pathways force regulators to look past the pest itself and evaluate the entire ecological network. The first major pathway is food web impacts.
Okay.
If we successfully eradicate the spotted-winged Drosophila in a region, what happens to the native wasps, the spiders, or the birds that rely on those flies and their larvae for food? Do their populations crash as a secondary effect?
So we risk starving the predators. That's a massive issue. But what if the weapon itself escapes its target? Like the second pathway they outline is hybridization.
Yes, hybridization is a huge focus.
Because there are roughly 2,000 different species of Drosophila globally, many of them are completely harmless or even beneficial to the environment as decomposers. What if the lethal gene drive jumps from our target pest into a closely related, harmless cousin species because they accidentally crossbreed in the wild?
That is a primary concern for risk assessors. And the third pathway takes that fear even further, which is horizontal gene transfer.
What does that mean?
This is the extremely rare but scientifically possible event where edited genetic traits could transfer to entirely different organisms outside of the target species' immediate family tree.
Like skipping species entirely.
Right, perhaps through a shared parasite or a virus.
Right.
When you introduce a synthetic genetic construct that is actively designed to cut and paste itself into DNA, you have to definitively prove it won't somehow copy itself into a non-target species.
Which perfectly explains why the scientific community isn't unified in just slamming the go button on this.
Not at all.
I mean, we have a source from Taylor and Francis literature explicitly calling for a, quote, pause and independent reevaluation of even the Wolbachia releases for dengue control, let alone full synthetic gene drives. Why are they calling for a pause on a natural bacteria?
Well, the authors of that paper raise concerns about unforeseen evolutionary pressures.
Okay.
Even though Wolbachia is natural, by artificially infecting a massive population of mosquitoes, we are applying a sudden, intense selective pressure on both the mosquito and the viruses they carry, like dengue.
So they might adapt.
The concern is that we might inadvertently drive the evolution of a more resilient mosquito or a more virulent strain of the virus that finds a way to bypass the Wolbachia firewall entirely.
Oh, wow.
They argue that the ecological and epidemiological long-term data just isn't there yet to justify mass, unregulated open-air releases.
So what does this all mean for you, the listener? I mean, you want to protect your local agriculture from invasive pests that drive up food prices. You want your family to be safe from devastating diseases like dengue and Zika. But you also definitely don't want scientists to accidentally break the global food chain or spark an uncontrollable genetic event. If a technology is literally designed to bypass normal biological containment, how do regulators safely test it to ensure these pathways to harm remain closed?
This raises an important question, and it is perhaps the most difficult hurdle in the field of synthetic biology today. The Oxford paper emphasizes that regulators require a strictly stepwise or phased approach to testing.
Okay.
You never go straight from the petri dish to the open field.
So where does it start?
Step one is locked, highly secure laboratories with multiple layers of physical containment. We're talking airlocks, specialized filters, and strict decontamination protocols.
Get it, Ben?
Once the genetic stability is proven there, step two involves moving to physically and ecologically isolated environments. For example, researchers might conduct the first outdoor releases on a remote island.
Because the insects physically cannot fly across the ocean to the mainland.
Exactly.
Yeah.
Or, alternatively, you conduct the trial in a geographic region where the winter temperatures are so harshly cold that if the modified insects were to somehow escape the test zone, they would immediately die off.
So they couldn't establish a permanent breeding population.
Right.
But how do they track them during these tests? Like, if you release 1,000 mosquitoes on an island, how do you find them again to see what happened?
Scientists rely on built-in genetic markers. Often, they will engineer the mosquitoes to express a fluorescent protein so they physically glow under a specific UV light.
Oh, that's clever.
Yeah, it allows field researchers to trap insects and precisely trace the movement, lifespan, and reproductive success of the modified insects in the environment.
Okay.
Only after years of this highly controlled, stepwise data proves that the pathways to harm are negligible do regulators even begin to consider an open field release on the mainland.
It is a remarkable progression to think about. Over just a few generations, we have gone from swatting bugs with rolled up newspapers to blanketing cities in chemical fogs to laser sorting Wolbachia saboteurs and now to rewriting the genetic code itself to create self-destructing pests.
It really is.
I mean, we are standing on the threshold of an incredible era. Diseases that have plagued humanity for millennia and pests that have routinely threatened our global food supply might soon be eradicated not by brute force, but by microscopic biological engineering.
It is a profound shift in our relationship with nature. We are shifting from attempting to control the environment from the outside to actively gaining the power to permanently edit the wild from the inside,
Right.
which means we carry the ultimate ecological responsibility to ensure the cure isn't worse than the disease.
And as we wrap up this deep dive into the sources, I want to leave you with one final provocative thought inspired directly by the Oxford paper. They briefly touch on the massive legal hurdle of transboundary movements and international protocols.
Yes, that's a huge issue.
Consider this. If a single mosquito carrying a highly efficient, low-threshold suppression gene drive is legally released in one country, it does not recognize human borders.
Obviously not.
If a storm blows that mosquito into a neighboring country, a country that never voted for this, that never consented to the experiment, and perhaps relies on a closely related insect for their own agriculture, and it begins eradicating a native species there, whose laws apply?
It's a legal nightmare.
How do we globally govern biological technology that, once released from the lab, literally has a mind and a drive of its own? Next time you swat at a mosquito, think about the invisible microscopic blueprints currently being designed to replace your hand. Until next time.